Okay, let's do section 4, proteins. Proteins are polymers that are made out of amino acids. Amino acids are the monomer, and they are bonded together by a special type of covalent bond called a peptide bond. If you were to dry out your cells, get rid of all the water, half of what's left would be protein. Scientists have identified several 100,000 different proteins and are constantly working to identify more. 2 or more amino acids joined together are called peptides. Whenever you have a long chain of those amino acids joined together, they are called polypeptides. Poly meaning many, peptides meaning amino acids bonded together. A protein is a polypeptide that has been folded into a particular shape, and that shape is essential for its function. So if you were to change the shape of the protein, it can no longer do its job correctly. Proteins have lots of different functions. They are used in metabolism, which is the sum of all of the chemical reactions in a body. Enzymes, which are a type of protein, act as catalysts to accelerate chemical reactions, meaning chemical reactions can occur much more quickly. Some proteins function in support, like keratin and collagen. They give cells their shape. Some proteins are used in transport. They are found in the cell membrane and allow certain substances in and out of cells. Hemoglobin is a very important transport molecule, a very important protein. It is the molecule that distributes oxygen to our cells. Some proteins are used in defense. They are antibodies that are part of our immune system. They bind to bacteria and viruses and prevent them from replicating in our cells. Some proteins are used in regulation that influence our metabolism, so they tell other proteins what to do. And some are used in motion. Microtubules, which are an important part of our cell's skeleton, are made up of microtubules, which are a type of protein. They are made of 2 different specific proteins, actin and myosin, and they contract and allow our muscles to contract. Amino acids are our monomer that make up proteins. There are 20 different ones, and they have the exact same structure except for this R group right here. This group right here changes, and depending on what it looks like, we have a different amino acid. And so here are the types of amino acids. We have valine, methionine, serine, lysine. There are 20 different ones, and we're going to go into more detail later on this year about the functions and the properties of those. Right now, I just need you to know that the only difference is this shaded part right here, and that there are 20 different amino acids. So here we have the synthesis and degradation of a peptide. So here we have our amino acid, and we bond it to another amino acid through the removal of a water molecule. So dehydration, synthesis is happening again. If we go back the other way and we stick a water molecule in there, we can break those bonds through a hydrolysis reaction. Remember, hydro means water, lysis means to break, so we are breaking that bond using a water molecule. The shape of protein and levels of protein structure. Proteins cannot function properly unless they are folded into their proper shape. When a protein loses its shape, it is said to be denatured. We can denature proteins through the exposure to certain chemicals, a change in the pH, high temperature, or some kind of mechanical means like whipping egg whites. That changes their shape, which changes their structure, changes their function. There are 4 different levels of protein structure: primary, secondary, tertiary, and quaternary. The primary structure is the linear sequence of amino acids. It looks like beads on a string. Hundreds of thousands of amino acids form a polypeptide, okay, but they're made out of just those 20 amino acids. So it'd be like having 20 different beads, different colors, different shapes, and we just string them all together. That's what the primary structure looks like. If we change the sequence of those amino acids, our structure is going to look different. Our primary structure is going to be different because we use different beads. The secondary structure occurs when that linear sequence, that long string of beads, starts to fold up into alpha helices, so basically like a twisted ladder, like a helix, or beta sheets, which are like sheets. They're pleated, they're kind of folded a little bit. And these are held together by hydrogen bonds. The overall three-dimensional shape of the polypeptide, once it finishes folding, is called the tertiary structure, and it is stabilized by the presence of hydrophobic interactions, hydrogen bonding, ionic and covalent bonding. So secondary structure is whenever it's first beginning to fold and it starts taking on an overall shape, and the tertiary structure is really due to the hydrophobic and hydrophilic interactions. So the hydrophobic regions of our protein fold up so that they are on the inside and they never have to come into contact with water. The hydrophilic regions are folded so that they are on the outside so that they come into contact with water. And then our quaternary structure consists of more than one polypeptide, so more than one structure that has been brought together to form one bigger one. Kind of like when the Power Rangers come together to form the big, robot thing. That's kind of what I think of when I think of quaternary structure. Here we have a diagram. Here's our primary structure, where it's just a long string of amino acids. Here we have our secondary structure. Here's an alpha helix that has started to twist up. And here we have a beta pleated sheet. Notice those can come, different regions form the alpha helix and different regions form the beta pleated sheet. And whenever they start to fold up together, that forms our tertiary structure. And then our quaternary structure forms whenever we have more than one polypeptide chain that has been folded working together. Depending on the shape of our protein, we get different outer looks. So my hair would be this long, straight hair that doesn't have a whole lot of texture. Can't do a whole lot with it. Usually a ponytail is about all I can do with it. But my roommate in college had hair like this beautiful, curly, thick hair that you can do a lot with. So if we looked at our hair structures under a microscope, mine would look like this, whereas Lizzie's would look like this. And here we have a spider web. Spider webs are made up of proteins that the spider excretes in a specific way to form its web. So the importance of protein folding and protein folding diseases. Chaperone proteins are proteins that help other proteins fold in the correct way. Defects in chaperone proteins may play a role in several different human diseases such as Alzheimer's and cystic fibrosis. Prions are misfolded proteins that have been implicated in a group of fatal brain diseases known as TSEs. Mad cow is an example of this. Prions are believed to cause other proteins to fold the wrong way. So once you get a prion's disease, it just causes a whole bunch of other proteins in your body, other than the original one that has been affected, to fold incorrectly, causing a whole myriad of problems. And that's it for this section.